Welcome back, everyone. In this video, I'm going to go over the practice PAT for environmental chemistry. Now, as I've said before, make sure you do the questions first and then follow along with me to see if there's anything you need extra practice on. Okay, number one here. So we want to know which of the following substances is inorganic. Now, keeping track of that definition can be a little tricky, at least for me anyway. So I like to think about it as what substances are organic first. So organic molecules are going to contain the following. Often, if it's organic, it's going to contain for sure a carbon and a hydrogen, and then sometimes an oxygen. So looking at these molecules here, what I've done just to save time is I've broken down the molecules. So here's what they all look like. So you can see any of those, it's kind of an odd way to think about it, but if we're eating food and it often tastes really good, unless if you're maybe allergic to peanuts, most time if it's sweet or if it's something like bread, it's going to contain what's called carbohydrates, and carbohydrates are going to contain these compounds, which would make them, chemically speaking, organic. So that means the only one that isn't organic in this question here is going to be table salt. Okay, number two here. So with number two, we want to know which of the following statements best defines the term LD50. So honestly, with this one, what I'm going to do is right here, I'm going to put the definition for what LD50 is, and now we just need to match it up. So with LD50, we're saying it's basically you give a sample population a certain substance, and it's going to kill the dosage. The amount is going to kill half that population in the test population. So that means the answer for this one is going to be C. Okay, number three here. So I'm going to go ahead and read out everything regarding this experiment, and then we can use that information to answer the next set of questions. So line that up. The following 3 brands of fertilizers are administered to 3 seedlings in a controlled experiment. Before the administration of the fertilizers, each seedling had a similar root growth and 3 leaves. None of the seedlings had developed flowers. So that's going to be important for us to remember. Here's the composition of the 3 brands of fertilizer. So we've got our brands here, and we know they're made out of nitrogen, phosphorus, and potassium. And we will need to keep track of the values that are here. Secondly, we know that once we gave the plant each brand of fertilizer, after a 2-month period, this is what we noticed, right? So depending on the brand, certain plants exhibited more flowers, certain had longer lengths of roots, and another one had more numbers of leaves. Okay, number 3 wants us to know what is the manipulated variable in the experiment. So I've said this before, manipulated variable means what's the one thing we change in the experiment, right? And I've used that analogy where I say, in order for someone to be manipulated, you're trying to change the way they think. In this case, we're just trying to change the one thing in our experiment. So if we look at this overall, the only thing that we're really changing is the type of fertilizer. And I know that's confusing because we do have different compositions for each fertilizer, but that's what makes each fertilizer unique, right? So therefore, in order to answer this question here, it is going to be the brand of the fertilizer that's used on each plant, and that's because each brand contains a different composition of elements in it. Okay, number 4. So what I did with this one is I'm going to use a split screen, and that will give me half the page where I can talk about the information that's in the lab, and then the other half is answering the question. Okay, so number 4. Which of the following conclusions is supported by the results of the experiment? So a conclusion, more or less, is the outcome of the experiment. So I'm going to look at these options, and right now the first thing I notice is we're talking about the elements. We're looking at what nitrogen actually promotes, and we're looking at phosphorus or potassium. So let's analyze each one of these, and we're just going to analyze the first half so we can eliminate some of those options. So if I'm looking at it, the first one talks about nitrogen. So with nitrogen, the brand that has the highest content of nitrogen is actually going to be brand Y. So brand Y has our highest content of nitrogen. So now we can look at the outcome here and say, well, okay, with brand Y, which again had the highest content of nitrogen, what was the biggest outcome that we saw, or the largest outcome? So out of these numbers here, using that high content, it looks like the number of leaves were impacted the most. So basically, if I were to use fertilizer Y, I got more leaves on the plant. So now I'm gonna look at my options and cross off anything that doesn't belong there or doesn't support what I just said. So nitrogen promotes root growth. We can see A and B are not going to be valid options because that's not the case. Now our next option then leaves us with looking at potassium. So with potassium, the highest one of potassium, I'm just gonna change my color here, sorry, is brand W. And brand W, if we look at what its outcome was, it gave the highest number of flowers. So now we need an option that indicates it gave us the highest number of flowers. So out of these two options, only one of them talks about flower growth, so that's going to be option D. Okay, number 5. So we've talked about our definition of organic molecules. So let's just go through this one really quickly. So we want to know which of the following is organic, meaning this time we're looking for the carbon, hydrogen, and oxygen. Silicone is just an element on its own. That's not going to work. Same thing with sulfur. So then we can notice a pattern. The PAT does want you to recognize the difference between salt and sugar. We know salt is sodium chloride, and we know sugar is going to contain our carbon, hydrogens, and oxygens. So our answer for this one is going to be C. Okay, numerical response number 1. So this is handy. They gave us a pH scale. Now, one of the questions I got in the comments of the videos is that we're looking for tips. So this is a great tip. Often they will give you something in the test that you can use again and again. So throughout the video, I've been flashing the pH scale. We can use this. It'll just be your job to recognize that this is an acid, this one here is neutral, and this one's a base. So now let's use that scale to answer the following questions. So we know when we have our lime juice, there's no color change and the litmus paper turns red. Well, we know that means that this is going to be an acid. When we look at ammonia, ammonia is pretty high on that pH scale. So we know that this is going to be a base. With water, we know that that's going to be neutral. Liquid W, okay, so liquid W is turning red. Well, where have we seen this before? We've seen it before up here where our litmus paper turned red. So in that case, we know that this one's going to be an acid. Let's look at X. Liquid X turns blue. Again, where have we seen that before? We've seen it here. So we know that that's going to be a base. And sorry, one thing I wanted to mention was with litmus paper, when it turns blue, it is indicating a base. So you can think of blue base, if that helps. Liquid Y, there's no color change, but it does turn red. And again, we've seen that before, so that's probably an acid. And our last one, no color change. We've seen that really clearly here with our water, so this one is probably neutral. So now let's put those in our numerical response bubble sheet or online, however you're doing it. So liquid W, we said, is an acid. So anytime we see an acid, we're going to pop a 1. Liquid X is a base, so we're going to put a 2. Liquid Y is an acid, so that means it's going to get a 1. And our last one is neutral, so it's going to get a 3. So our answer for this one is going to be 1, 2, 1, 3. Okay, number 6 here. So number 6, we want to know which of the following rows classifies the type of data that were collected during the study and identifies the type of monitoring that was conducted by experiments. So let's look at our graph first. So here's our graph, and we can see on the x-axis, we have the pH of the lake. One thing I do want to point out is they were a little sneaky in this. They put the neutral pH not in the middle where we often see it when we're looking at pH, but they kind of stuck it to the side. So keep that in mind. And then the other thing is on the y-axis, they gave us the number of organisms. So if any of you all did kind of a field trip with this unit, this is what you did. You went to the water source and you looked at what's called bioindicators. You looked at molecules that would be in place if the water was healthy. Then you went a little further down the river, wherever it was that you went, and you collected animals and samples again, and you look to see how that information changed. What you're doing is you're looking at bioindicators. Essentially, the healthier the water, the more certain type of specific organisms will live there, indicating that the water is pretty healthy. So let's keep that in mind to answer this question. So we want to know, out of these, what kind of information did we collect? So the first thing is the classification. We want to know if it was qualitative or quantitative. So if it is quantitative, that means we're actually collecting numbers. And if it's qualitative, you're looking at kind of basic information indicating the quality of something, okay? So for this one, we can see on our graph, we are definitely collecting numbers. So that means right away, I can cross off the qualitative ones right here. Our second piece of information wants to know the type of monitoring. So of course, we're testing the number of organisms, so that's going to be a biological type of monitoring. Therefore, our only answer for this is going to be C, as we're not actually doing any chemical monitoring. So number 6, the answer is going to be C. Okay, number 7. So this goes back into play of what I was talking about in keeping track of that neutral pH. So here is our neutral pH. And for number 7, what we need to do is decide which species has the greatest number of organisms present when the water is neutral. So neutral again is going to be around 7. And so now what we need to do is we need to read our graph and we need to say, well, okay, here is our neutral pH. Well, which organism has the highest? So I'm going to go ahead and use my ruler here. And I'm going to say, well, okay, there's only really one organism that's hitting that high point at neutral pH, and that's going to be the one with all these dots here. So that one to me looks like species B, which means our answer for this question is going to be B. Okay, number 8. So with this question, we want to know if acid rain falls into the lake and changes the pH of water from 6.3 to 5, then the species that is expected to increase in the number of organisms is blank. So looking at our graph, this one is a little tricky, so I'm going to walk you through this. So looking at our graph, we need to notice pH 5. So our pH of 5 is going to be here. And I want to highlight 2 organisms that tend to be thriving. So the first is the organism represented by the solid line. I believe that is going to be species D. And the second organism is this one here. Now, what we need to be careful is what the question is asking us. So it's asking us, as we add more acid rain into the water, which species is going to continue to increase? So with species D, which, I think would be tempting to say is the answer, species D, yes, it does survive in a pH of 5, but if we notice, it also still survives in a pH of 6.3. Both those pHs, we can see a consistent amount. So what that means is while that species will be found in both pHs, it's not actually going to increase in population size. So that means species D isn't an option. We know we're not going to choose species A or B because those ones really only start to increase in our kind of neutral pH. So that only leaves us with leaves us with species C, and that's because species C will thrive in a pH of 5. So as the water goes from, as the water gets more acidic, these guys will have a better chance to survive. So our answer for this one is going to be species C. All right, we've got number 9 here. So number 9 wants to know which of the following roles is played by carbohydrates in nutrition. So I just want to be clear here because carbohydrates have such a bad rap. Everyone often thinks that they're just bread, which is great. I mean, they are bread, but they're not only bread. They're also fruits and vegetables. So the key thing with carbohydrates is they actually provide us all our energy source. Without these, we actually can start to feel pretty weak. So these are going to be ones that goes back to that organic compounds. They're going to contain our carbon, hydrogens, and oxygens. But that also means that our body can digest it quickly and provide us with a good amount of energy. So our answer for this one is going to be C. Hey everyone, I just wanted to say thank you for all the love and all the great comments that were coming my way while you were practicing for PATs. Just as a reminder, I am also here to help you in grade 10. So make sure you reach out, make sure you let me know what you're struggling with so I can make a video that's gonna help you. All right, number 10. So we have our pH indicator here, and they've given us some information. What they're saying is if it's above 7.6, it's going to be blue. Anywhere between 6 and 7.6, it's going to be green. And finally, anything below 6, it's going to be yellow. So what we need to do is decide when we use our indicator and it's going to be yellow, really what substance are we looking at? So we need to be really clear about our pH scale here. And I'm just going to go ahead and write in what we would expect at each kind of color. So if it's above 7.6, we're going to be looking at a base. If it's between 6 and 7, we know we're looking at something that's neutral. And if it's below 6, we know we're looking at an acid. So if all of a sudden we mix it and we get our outcome to be yellow, so that means we're looking at something that's going to be in this range here. So that means we're going to be looking at an acid. So our answer for this one is going to be B. All right, number 11 here. So with this one, we want to know what volume of solution B must be used to neutralize solution A. So we got to think about, first of all, what does it mean for something to be neutral? Well, we know neutral is going to probably be around 7. And then we need to know how to use or read this graph. So in this graph, we have our pH. So we've got our acids here and our bases here. And like I said, neutral is in the middle. And then we've got our volume of solution B being added to solution A. So what this means is we started off with something acidic, excuse me, and we kept adding to it. As we added to it, the volume started to increase, but so did the pH. Meaning, since we ended up about here, we probably added a base, probably I would say a pretty strong base, to a strong acid to neutralize the two. But we want to know which volume did that neutralization occur. So for something like this, I'm going to use my ruler again, and I'm going to say, well, okay, pH 7 is right around between 6 and 8. And I'm going to use my graph and draw a line and let it hit that line. Once it hits that line, I can rotate my ruler and go all the way down to get the volume. So that means I'm looking at a volume, again, between 20 and 30. So the only option that works for me here, a number between 20 and 30 is 25. So our answer for this one is B. Okay, number 12. So number 12 says the number of mayflies decreases as the habitat quality of a stream declines. So with this mayfly, we're looking at what's called a bioindicator. So what this means is the more of this organism you have, especially a mayfly, the more it means you have a healthy environment because they're not going to thrive in an unhealthy water quality. So for this one, we're actually using something called bioindicators, which means really we need to decide, keeping that in mind, we need to decide the following. The study of the number of mayflies can be useful to scientists for blank: chemical monitoring, biomonitoring, estimating the concentration of lipids in the tissues of a particular species, and estimating the concentration of toxins in the tissues of species. So C and D are interesting options. The only thing is we're only looking at the number of mayflies. We're not looking at how much toxins or lipids were involved in a series of organisms. So those ones won't work. And we're not really looking at any chemicals here. We're just looking at, again, the number of bioindicators. That means our answer for this one is going to be B. Okay, number 8. The dissolved oxygen concentration of a pond is 8 parts per million by volume. So what this means is if you had 1 million milliliters of water, 8 of that would be the dissolved oxygen. So this indicates that the level of dissolved oxygen, that 8 milliliters, is found in blank. So essentially, if I were to write it out, just so we're clear, we're saying it would be something like this. So then that means the only option for this that's going to work is going to be A. All right, number 14. So Tanisha was curious to see how adding water to vinegar would affect the pH of a particular solution. She set up the following experiment. So here's our beakers. And what she did is in the first beaker, it was 100% vinegar. And we can see as she kept going, she decreased the amount of vinegar. Which, by the way, this is a great cleaning solution, especially with right now and our water restrictions. This works really good to rinse off your plates, and then you can stick them in the dishwasher once the dishwasher load is full. But let's actually answer the question. So we want to know what is the manipulated variable in this experiment? So again, we want to know, well, what is she changing? So in this experiment, the only thing that she's changing is the ratio of vinegar to water. So we need an option that's going to fit that. So the option that's going to work best for this one is going to be option D. Okay, numerical response number 2. So we've got our graph, and let's read the information that's at the top. So it says here, a food chain is a linear representation of the feeding relationship between organisms. Plants are often the first species shown in the food chain. Large meat-eating animals tend to be the last species shown in the food chain. A biologist studies the concentration of mercury in the liver of 4 species that are in the same food chain. She records her findings in the graph below. So one thing I want to be clear about it is we're looking at the concentration of mercury. Now, mercury is one of those toxins actually responsible for something called Mad Hatter's disease. And essentially what happens is through the food chain, we get something called biomagnification. So what happens here is a small organism gets exposed to the toxin, but the organism that eats it gets exposed to the toxin a little bit more because it had to eat more of the little organisms. So it starts to build up in the food chain. So what this means then is looking at our species, we want to know which one would be the first in the food chain and which one would be the last in the food chain. So as I said, it starts to build up. That means this organism here that has the highest concentration of mercury is actually going to be at the end of the food chain because they had to eat lots of organisms to survive, and all those organisms had to eat other organisms, and the toxin was able to magnify throughout the food chain. The species that has the least amount of toxin would be first, and then we can just put them in order just like this. So our answer for this one is 3, 1, 2, 4. Number 15. So we want to know a solution can be classified as an acid when its pH is blank and can neutralize blank. So if we know we're looking at an acid, we're looking at something that's going to be less than 7. So I can cross off these 2. And if it's going to neutralize something, so if you have an acid, it's going to neutralize the opposite of that. So that's going to be a base. So that means our only option that's going to work for this is going to be C. Okay, number 16. So they gave us a pretty interesting chart. They said here are some of the substances that are essential to basically living and growing for all healthy things. We've got our organic substances, which I'm hoping by now it's in your brain and you're thinking carbon, oxygen, hydrogen, and our inorganic substances, which just means it doesn't contain carbon, oxygen, hydrogen. So from this, we need to decide which of the following would fit the table below. So we've got our vitamins, we've got our lipids, and then we have our eggs and our nuts. So our eggs and our nuts are really, when we think about the nutritional food group, it's really going to be our proteins. We have our grains and our fruits, and this is the one that I was talking about earlier, where we tend to think of this group as just grains, but it does include our fruits, to give us a lot of energy. So this one's also going to be our carbs. And then finally, we've got our last one, which is iron and calcium. Well, that's really going to be our minerals. We need these to also survive. So the table that matches up this, the row that matches up this is going to be option D. Okay, number 17, we want to know which of the following actions most clearly represent biological monitoring due to environment quality. So with biological monitoring, we know we're talking about organisms that are going to be going through and surviving, indicating the health of the place that they're surviving, aka environmental quality. So I'm looking at these options, and option A says the number and diversity of invertebrate species in a pond near a wastewater treatment plant are measured over 10 years. So I really like this one because they're saying here's our wastewater plant, but we're looking at the species that exist there. And of course we know if there's lots of good species there, it means the waste that is being produced from that treatment plant is being carefully thought about so our water isn't being ruined. So I do like this option. Let's go through the rest. For B, it says the pH of a river is measured every 2 months for 5 years to track the impacts of a local industry. So that one is a really good one, but it's not talking about the biological monitoring. So that one's not going to work in the case of this question. Option C, the concentration of mercury underground near a waste disposal site is tracked over 2 years. So again, really good thing that we're doing that, but it's not using the bioindicators. And finally, option D, the population of geese in a wetland is measured and is compared to the population size 10 years ago. That one does look like it could be a good one. The only thing with this is it's just looking at population size. It's not actually indicating if that's going to impact the environment quality. So our answer for this one is going to be A. All right, last one for this practice PAT, number 18. One method used to reduce sulfur dioxide emitted into the air is to use smokestack scrubbers in industrial plants and electrical generating plants. So if we're not sure about this, what it is sulfur dioxide, or what they call in the chemical world SOx, combined with, and they didn't put it in this question, but just so you know, combined with NOx, which is nitrogen, and oxygen. And sorry, let me clarify, the X just means like the X in math, where that number could be 1 oxygen, 2 oxygens, or 3 oxygens. What happens is when these pollutants get into our atmosphere, they can start to combine with the clouds and create acid rain. In fact, in the description below, I'm going to leave a little funny video just so you can watch kind of how acid rain takes place, right? But let's go ahead and let's read the question. So we know that this is a method that we can use. Based on that information, the practice of using smokestack scrubbers would most directly result in a reduction of. Okay, so I indicated earlier that this was acid rain. So therefore, the only one that's going to fit this one is going to be option B. Hey everyone, I hope that was helpful. Just as a reminder, in the description, there's some interactive tools to help your learning. And I also hold one-on-one sessions. And just so you know, when you all are in grade 10, I will continue to hold those one-on-one sessions. So make sure you reach out if you need some extra support. Until then, everybody, I'll see you all on the next one. Chain, we get something called through the food chain, we get something called